Okay, folks, I think we'll get started. So thanks for showing up. It's a great turnout today. Thanks very much for coming. So it's my pleasure to have the opportunity to welcome Dr. Wilder Greenman to today's seminar. And there's no way I can do justice to his incredible CV and range of experience, but I'll hit some highlights, and I'll say it's been probably the greatest stroke of dumb luck that Wilder approached me in 2022, I think, after moving here to Halifax to inquire research opportunities here at Dalhousie. He's been with our group as a postdoc from 2022 to 2024. And then last year, Wilder identified this amazing funding opportunity from the Weston Foundation, the folks that own Loblaws and Superstore, the people that we give all our money to. Well, they're giving some of our money back to us and to Wilder. So Wilder's the PI on a research project that we'll talk about today involving deep water corals and food webs off Labrador. And as part of that, really interesting collaborations with the Enuiatin First Nations. Just as a little bit of background, so you know where he's coming. He comes from quite a different place from the research that he's doing now. So he obtained his bachelor's, a combined double major in biology and geology from the University of Ottawa, completed in 2015, and then from there he went to do his master's, also at the University of Ottawa, completed in 2017. And then his PhD at McGill from 2018 to 2012. Notice on time, on track and on time. No gaps or lengthy delays. And he worked at McGill for his PhD with Galen Halverson. And also Paul Hoffman, if you're familiar with that name, big name in the world of snowball Earth, one of the originators of snowball Earth theory. Did a lot of field work in Namibia to support that work. But some of Wilder's most interesting experiences in the far north, in the Northwest Territories and in Enuiatin, working on Precambrian sedimentology and some paleobiology implications. And if you ever have the chance to allow him to show you some of his slides and videos from working up there, it's stunning. It's some of the most amazing scenery you can imagine. And some really cool research has come out of that. He's got a huge publication list, first author publications in geology and the Journal of Sedimentary Research and scientific reports. And, so with that, doesn't do it justice at all, but hopefully that gives you a bit of an idea where he's coming from. So on to you, Wilder.
Thanks, Evan. So as I was preparing this talk, I kind of was getting sick of this kind of a bit jargony. So I switched it up to something that I think is a bit more descriptive of what it is in the terrain. So at the Makkovik Hanging Gardens, really we're trying to figure out what deep water corals and sponges are eating at this site and how this has changed over time. So I'm part of the Stabilizing Biogeochemistry Lab led by Owen, and so we use isotopes as a lens to understand biogeochemical cycling in the ocean. And for this project, we're really interested in understanding what food is making it to the deep ocean. And corals are particularly useful for this because of their skeletal makeup allows us to combine our isotopic work with sclerochronology to generate these long time series of how that food has changed over time. And so, as you can imagine, I spend a lot of time thinking about corals, and when I prepare a talk like this, I need to remind myself that that's not normal behavior, and that it's important to point out why we should actually care about corals. And so they actually provide a lot of ecosystem services. So we have these complex coral and sponge habitats, hundreds to thousands of meters below sea level off eastern Canada. And they provide critical spawning grounds, food, substrate, shelter for many species, including economically important ones. Unfortunately, these habitats are highly susceptible to destructive fishing practices, so they're very fragile. So you can see in this video here, even though this habitat is about 450 meters below sea level, you can see these large paragorgic corals, which can be 2 meters tall. They're enormous, but you see these long lines interacting with it even in the deep ocean. So they're not immune from human contact, and they're very fragile, so they can easily be destroyed by this type of activity. And on top of that, they're very slow growing, so if they are destroyed, it takes a very long time for these habitats to regenerate. So for these reasons, corals and sponges are considered indicators of vulnerable marine ecosystems, which makes them very important for identifying areas in need of protection, which fits into Canada's commitment to protect 30% of our oceans by 2030. So that identifying these habitats is a very important component of that. And for our part, we're very interested in understanding what it is that they're eating over time too, and see how this changes and what's happening in the water column to understand how threatened they are in some of these habitats. So now moving into the Makkovik Hanging Gardens. The Makkovik Hanging Gardens are located off Elucidating, which is a self-governed indigenous region in northern Labrador. And the Makkovik Hanging Gardens are a vertical wall community of deepwater corals. So the top of the wall is around 450 meters below sea level, but it goes down to close to 700 meters. So it's a large wall with a really impressive biodiversity covering it. And so this is well below the photic zone. And on top of that, these are relatively close to the shore compared to a lot of other coral habitats. So for much of the year, they're actually covered in sea ice. So it's actually an incredibly bleak habitat to try and live in. But when you go down there with a remotely operated vehicle, you can see that there's these beautiful biodiversity oases, so they're pretty special. Okay, so here is a video of multi-beam data that was turned into a 3D reconstruction, and it shows you the topography on the shelf here where these walls are occurring. So you can see these are transects that were done with an ROV, and different samples were collected, moving along to some of the different ones that have been done predominantly by DFO over the last few years. And so again, the height of these walls can exceed 100 meters or so, and you have this impressive biodiversity on them, samples taken all the way up. So when you go and look at them, they look something like this, where you have these large corals all hanging off the wall. So these Pernoa rosetiformis corals that we study here, these lasers are about 10 centimeters apart, so they're quite sizable animals. And then all these white blobs are sponges, and the closer you look, the more you see. So there's a lot of life on these vertical wall communities. And I think that the discovery of the Makkovik Hanging Gardens is a really nice example of the value of combining Western science with local indigenous knowledge. And that really starts in this area with the Makkovik plan, which translates to our oceans. So the Nunavut government is coming up with a comprehensive marine plan for the area, and a large component of this is finding these sensitive habitats and designating them for protected sites. A major challenge with actually identifying these sites is finding them in the first place. So you can see here we have this habitat suitability model that was published in 2017. And you can see in red, these are sites that are considered high habitat suitability for the large corals that we study. Now, it's way too large of an area to systematically look at with a remotely operated vehicle. And it's kind of like the marine equivalent of ground truthing and geologic contact, where we actually have to go there and look at it to see if that contact exists. And so what the Nunavut government did in collaboration with the DFO is work with fishermen in the area and cross-reference these sites that were high habitat suitability models, that fit the high habitat suitability models, and they cross-referenced that with fishing sites in the area where they had known net contacts. And so this paper here goes over that whole process, and the list of co-authors really highlights the collaborative effort here. So you have folks from DFO, you have folks from the Nunavut government, owners are co-author on this, and that sort of starts where we began our involvement with the Western Founding Foundation. So because this site was so recently discovered, we had some pretty basic questions about what was actually happening in this area, including how threatened these deep water coral and sponge habitats are. We're very interested in understanding what these deep water corals and sponges are actually eating, what does the current food web actually look like, and how have the diets of corals at the Makkovik Hanging Gardens changed over time. And so the lens that we look through that through is using isotopes, because it's actually very difficult to understand what these things are eating. You can imagine in such remote settings, you can't just go there and watch what they're eating. It's very hard to look at their gut contents because it's so small, and the food they're eating is so small. So we use isotopes to understand this problem. And here you have a view. I think we have this basic understanding of how marine food webs work. And here, I just want to highlight kind of what we're interested in here. So obviously we have particular organic matter is formed in the surface oceans and acts as a currency that percolates throughout the food web. We know that deep water corals and sponges at the bottom of the sea, they also rely on this particular organic matter. But at the deep ocean, only a small fraction of that actually makes it there. And so any small perturbation in the surface ocean can have large effects on the viability of these deep water coral and sponge habitats. And to visualize kind of some of the things that we're interested in, I asked ChatGPT to make this little view schematic, kind of representing the water column, like one of those sushi restaurants where you have a conveyor belt bringing the food around. So you can imagine that the surface ocean kind of acts like a kitchen, where this is where the food is made, and then we have the biologic pump here. So how food is actually making it to the deep ocean. The efficiency of this conveyor belt really dictates what food is going to make it there. And corals and sponges basically have this seat kind of right at the bottom, so they're left with whatever makes it to the deep ocean. And so we're very interested to understand how that kitchen is changing, what's happening in the surface ocean, and how the efficiency of that conveyor belt, that biologic pump, is working. So one example of why we're concerned is because we have one example here from researchers at Dalhousie that showed even drops in phytoplankton biomass in the North Atlantic. And so again, you can imagine that if there's less food being produced in the water column, we may have less food making it to the deep ocean. This is just one of many examples that we're seeing in different areas. And we see response from certain animals to this as well. So things like lobster fisheries are moving north generally because the lobsters are unhappy in their old habitats and are moving, but corals and sponges are sessile organisms. They're kind of forced to stay where they are and rely on the food that makes it to them. And so I'm going to give a case study from work that we published earlier this year, and it's going to do three things. So it'll outline the principles of compound-specific isotope analysis. This is the primary method that we use. I'm going to describe how we study food webs using this method, and I'm going to go over what we're learning about them and how it's dictating what we're doing, and what questions we're asking at the Makkovik Hanging Gardens. So here you can see our sample site for this case study is Shortland Canyon, which sits at the shelf right here off Nova Scotia. And when you go there with a remotely operated vehicle, as Owen did in 2022, you see a site that looks like this. So you have these large Paragorgia arborea corals, again, up to 2 meters tall. You have these Primnoa recidiformis corals that are often about 1 meter in size. Keratosis gray, so these are the large corals that we're interested in studying. But there's a huge amount of biodiversity here, so there's also sponges and anemones, various different things. And so the point of this video is that we collected samples from this very constrained area. So the assumption being that their food availability should basically be the same at this site. And we wanted to see if they were basically eating the same thing. So our sample set represented different corals as well as sponges, ascidians, and sea anemones. So we had this wide range of different benthic invertebrates that would tell us kind of what represented the primary consumers at this site. And we started this study the way that a lot of marine food web studies begin. So we use bulk carbon and nitrogen isotopes. It's a very commonly used method. It's relatively cheap. You can get a lot of data quickly. And there's a lot of published data to compare it to. And so bulk...
Which one is sea anemone?
Hermathidae here is the sea anemone.
Okay.
Yeah. So often when these food web studies are done, we're mostly focused on nitrogen. And because nitrogen is used as a proxy for trophic transfer in these food webs. So in an example like this, you have things like particulate organic matter, you have large octocorals, sea anemones, and ascidians. But then we had this anomalously high value for our different sponges. And again, if you use this as a one-to-one proxy for trophic transfer, you would assume that these sponges are sitting almost like predators in this area. And when you look at the literature, there's actually a lot of examples of sponges exhibiting these really enriched delta 15 nitrogen values. And to put into perspective how high these values are, if you look at a similar type of marine food web study, you see a range in delta 50 nitrogen values of about 14 per mil that goes all the way from particulate organic matter up to polar bears. So we have a similar amount of enrichment in our study, but we're not trying to claim that sponges are predators in this site. Really the point is there's something wrong with the way that sponges behave using bulk isotopes, and we wanted to probe further into that question using compound-specific isotope analysis. And so some of the limitations of bulk δ′⁵ nitrogen is that we often assume that enrichment is kind of a one-to-one trophic transfer process. So that means that when we go about calculating trophic position, we take the δ′⁵ nitrogen value here, so the y-axis, and we make an assumption about what that food source is. In most cases, that would be particulate organic here. Here we would assume that that's what's making it to the deep ocean. And then when you go about calculating trophic position, things like sponges will get these really inflated values. The challenge of doing it this way is that it's really hard to account for baseline nitrogen, which is basically the nitrate at the base of the food web making it to that food source. So if these things aren't all eating the same thing, we can't really compare trophic positions for them. Another thing that's missing for this is the influence of microbial resynthesis. So as food falls through the water column, it can be resynthesized by microbes, and this can also enrich δ′⁵ nitrogen values. But when you look at bulk isotopes, basically you have all these processes into one value, and it's hard to disentangle. So when we think about organic matter that we're doing this analysis on, what makes it down to the deep ocean is already mostly made up of proteins. So proteins broken apart into amino acids, and so in our lab we break apart this organic matter into the individual amino acids, which we know fractionate in predictable ways. And then we can study those independent amino acids to get a more nuanced view into the different processes that go into this combined bulk signature. And so with the same type of organic material, instead of one data point, we get multiple data points spanning the different amino acids that go into that protein. So there's different groupings here. So we have the trophic amino acids. So we see trophic amino acids, they do become enriched when you have trophic transfer, and this is why nitrogen values do go up when you have that. We also have source amino acids. So source amino acids do not become enriched with trophic transfer, and they lock in that baseline nitrogen signature, which is really valuable for us because we no longer need to assume what their initial food source was. Which means that when we go about calculating trophic position using amino acids, we actually have it internally normalized by our trophic amino acids, normalized by our source amino acids, which is a much more precise way to calculate the trophic position because you no longer need to assume what that food source is. We also have a measure of microbial resynthesis, so we call this the sum V parameter. This is a qualitative measure of basically how scattered the amino acids are, the trophic amino acids are. The principle behind this is that based on culture experiments, we see that individual microbes tend to preferentially select certain amino acids. So we wind up with these jagged patterns within our trophic amino acids. That would look something like this. So this would be a high variance example. This would result in a high sum V value. And we would consider this to be a more degraded food source because it's been more heavily resynthesized by microbes. In contrast, we might have a much flatter line here. So for something like this, we would assume that microbes have not eaten it as much, and we would call this a fresher food source. So by doing this method, we can take these different processes that go into our bulk signature, and we can break them apart and look at them individually. So this gives us a much more nuanced view of what's actually going on with the nitrogen systematics. When we do that for our samples, we get something that looks like this. So this is the type of data that our lab absolutely loves, but I'll kind of explore some of the really key findings from this. So the most important thing that we saw in our bulk data was that sponges have these anomalously enriched values. And so if you look at the sponges here, so they're the orange and yellow, we can see that they have these really enriched source amino acid signatures. So pretty much right away we thought, okay, this has nothing to do with enhanced trophic enrichment. They're just eating something with a very different source amino acid signature. And if you go in a little bit deeper here, again, just highlighting the sponges as well as ascidians, which morphologically act very similar. You see these very scattered trophic amino acids. This is telling us that this food has been heavily resynthesized and we would consider this to be a very degraded food source with an enriched source amino acid signature. In contrast, if you look at the corals and sea anemones, you have much lower and tightly clustered source amino acids. You have relatively flatter lines. So this is kind of one similar food source that we would consider to be relatively fresh compared to our other examples. So by doing this, we're able to parse apart a lot of basically differences in resource utilization at a site where food availability should have been identical, we would think. And again, this issue that has persisted in the literature where there's these really elevated bulk values for sponges, we see it's pretty clear that it's probably because they're eating a very different food source. So viewed a different way, I have a different plot here. So along the x-axis, you have our SEMV parameters. So this is the microbial resynthesis. So further to the right implies a more degraded food source. Further to the left is fresher. And then on the y-axis, we have trophic position. So the trophic positions are all between 2 and 3. They're all primary consumers, slightly different diets, but not a huge amount of variety here. And now that we have looked at sponges and ascidians using complex physical isotopes, we see that they don't have this elevated trophic position anymore. They actually slot in like really nicely with this continuum of our different primary consumers. And so one of the questions we had at the end of this study was, okay, well, if they're not eating the same food, if they have such different source amino acid signatures, are we seeing that possibly there's one food web with surface-derived food material, which we assume to be the case for corals in this area, and then maybe the sponges are tapping into a completely different food source that's degraded, something kind of like dissolved organic matter. And so we went about testing this using δ′⁵N carbon. And so we had 2 δ′⁵N carbon curves that spatially bracket our site. So Shortland Canyon is here, and we had profiles from here and here. And basically we wanted to look and see if the values that we saw in the DIC mimicked the ones that we saw in our tissue samples. And so the idea here is that if corals and anemones are eating surface-derived food, it should have δ′⁵N carbon values that mirror the surface δ′⁵N carbon. So they should have, we would think, values around 5 to 20 per mil, and then diets rich in older reworked dissolved organic matter should have depleted, really old radiocarbon signatures, because dissolved organic matter in the ocean can be hundreds, if not thousands of years old. So we started by doing this for our cnidarians, and our δ′⁵N carbon values basically plotted close to those of surface values. So this is what we were hoping to see. This was a positive initial result. And then we started looking at our sponges and ascidians. And so for Hexaptonella, this glass sponge, we saw exactly what we were hoping to see. This really depleted value telling us that it is probable that it's a very degraded dissolved organic matter that's been floating around in the deep ocean for quite a long time. And when we looked at our other sponges and ascidian, they didn't really behave as well. So they returned more surface values. So this told us that on top of all the resource partitioning that we've seen so far, there's actually more different niches in the area. So it goes to show that even when they're all in the exact same area, you can have these very large differences in the types of food that they're eating. Okay, so for the takeaways from this, I hopefully have conveyed that common-specific isotope analysis is super useful for complex food webs, particularly when we have sponges which just don't behave when we use bulk isotopes. And again, even when food availability is the same, they can have very different source diets. A couple of things to note. There's very high selectivity from some of our corals, which is, we'll come back a little bit later. And what we think is going on here is that we have a high degree of complementarity rather than direct competition. So these things, the fact that there's different food sources coming in here is a good thing for supporting biodiversity in the area. And then the last point, this idea that it's possible that certain sponges are tapping into these really recalcitrant pools of food, is this idea that I've become quite interested in looking into deeper. And so it's not new that people think that sponges are eating dissolved organic matter. Here's 2 very relevant papers that describe what they call the sponge loop. So basically, sponges sit in these habitats, they tap into these dissolved organic matter pools, and they put it into their biomass, and then they pass on these nutrients to other organisms through shedding of spicules or even direct predation. And so this is a really big deal at a site that is exhibiting diminished particulate organic matter fluxes. If there's less food coming down to the water column, maybe there's this alternative pathway that's bringing in food into these deep water habitats and helping to sustain them. And so one limitation of studies like this, these are mostly lab experiments, and so kind of at the end of both of them, they say, we need more in situ examples to demonstrate that this is actually happening. And so we wanted to explore this idea at the Makkovik Hanging Gardens. And so there's kind of 2 points to demonstrate that the sponge loop is taking place there. So first off, can we actually find strong evidence that dissolved organic matter is being taken up by sponges, and are they passing it on to other organisms in the area? I'm going to focus a little bit on the second question here. So this is gets into Naleen's honors project. So her and I are working on this question. And so here's some of the very early results. So we still have to get more data for this, but what we're seeing is actually patterns very similar to what we saw from Shortland Canyon. So in blue here you see our corals, and in red you see our sponges. And similar to last time, we see more scattered amino acids, trophic amino acids for the sponges, and much more enriched values for the sponges. So it's good that we are seeing this pattern in another area. It's nice to see this repetition. The thing that changes is that we were able to also incorporate some of the potential prey. So we have sea star samples that are things that could be eating the sponges and corals. So that's what you see in pink here, pink and purple here. So first off, we have the Hippisteria sea star. So that's this guy here. And basically you can see they have much more enriched trophic amino acids because they're secondary consumers, they're eating this type of food. So that's good. And then you follow down the source amino acids, and for Hippisteria, it mimics that of the corals. And this is telling us that they're probably part of the same food chain. Whether or not it's directly eating the corals is not, we can't state that explicitly here, but they're eating kind of the similar food chain. Then we have our Tremaster mirabilis, these 2 upper lines here. So basically, again, they have these enriched trophic amino acids, but their source amino acids mimic those of our sponges at the site. So this is really exciting to demonstrate that we possibly have these 2 divergent food chains going on at the deep waters of the Makkovik Hanging Gardens. And so viewed a different way, here's another plot where you see phenylalanine on the x-axis. So this is our source amino acid. So again, sponges have high source amino acid values here. So further to the right, it's telling us that it's this enriched source amino acid. Further to the left is more depleted. So we have Primnoa, our corals on the bottom left here, and sponges on the bottom right. Very different food sources. And then if you look at the y-axis, this is our trophic position. So one trophic position above our corals, we see the Hippisteria sea star. So again, good evidence that they're eating as part of the same food chain as these Primnoa. And then Tremaster mirabilis, our other sea stars, again, not perfectly overlying it, but much greater affinity to the sponges. So this is telling us that maybe we have, again, maybe we're seeing these divergent food webs, one driven by particulate organic matter and one driven by dissolved organic matter. So returning to this analogy, it's kind of as if sponges and corals are sitting kind of at the bottom of the restaurant here. They're waiting patiently for the food source to come. And then sponges just like take out their own bag of snacks and start eating it. Don't wait for anybody. They have this alternative pathway to get food to the deep ocean. And not only are they eating it, but they seem to be passing it out to other organisms like the sea stars. And again, to reiterate the value of this, if there's lots of food coming down the conveyor belt, that's useful, but maybe kind of annoying. But if there's a diminishment in the food coming down the conveyor belt, suddenly that sponge is going to be playing an outsized role in this habitat if it can supply food to other organisms and tap into recalcitrant food sources that nobody else really seems to want to eat. And so this is kind of our isotopic line of evidence to support the sponge loop hypothesis at the Makkovik Hanging Gardens, but we also have video analysis that Naleen is doing. And so here's an example, one of many, where you basically see these sea stars, again, these are 10 centimeters, these sea stars sitting on these sponges. And there are countless examples of this. I asked Naleen basically, hey, could you show me a couple photos where there are sea stars sitting on the sponges? And she came and she said, I had to stop. They're just all over the place sitting on these sponges. And so basically, not only do you see them sitting there, but you often see these little bite marks within the sponges. So there's quite a strong case for qualitative spatial assessment there, but we're also doing more quantitative assessment to actually demonstrate that the sea stars love hanging out around certain sponges, and again, finding support for the sponge loop hypothesis. We're starting in situ. And so far, what I've been talking about is all the present. It's one basic snapshot of what the food web looks like at a time when those samples are collected. And so in this case, we're looking for isotopic variability between different specimens to see if their diets are different. But we can also look for isotopic variability over time. And in doing this, we look at the coral skeletons, which produce these annual growth bands. And so the organism is not changing, but each year their source diet could be changing. And we want to see if there are changes to that. And so the samples I've been showing so far are ones that were collected previously from last couple of years. But we really don't have any older colonies from the Makkovik Hanging Gardens or the Sentinel, which is a similar site that was actually discovered even more recently. And so this summer we went on the Amundsen, and we were collecting coral samples and various other samples to basically fill in some of the geographic blind spots that we had to understand the coral distribution and understand their diets within this area. And so how we work on these, here's a view, top left here, of the remotely operated vehicle coming in and taking a sample. So again, lasers are 10 centimeters. This is our coral of choice, Primnola zetiformis. Then you see this sponge is next to it. Our view comes in. And that's our sample. And so this was video taken from this past summer. We actually don't have these samples at the moment. Lauren O'Reilly, who was with us, is in Quebec City currently for demobilization of the Amundsen. So we're going to get the samples we collected next week, basically. However, this is one that I previously worked on. So you can see we have the whole colony here. And probably the best way to think about this is they grow very similarly to trees. So if you think of a tree, they have growth rings that they develop annually. And so if you want to capture the full lifespan of that tree, you would go to the base of the trunk. And so that's what we do with our corals. So this is the base of it. It's been growing outwards for its entire lifespan. And we want to get a cross-section right from the base of the trunk, which is why we're collecting these whole colonies. Here you can see what that looks like. Hard skeleton, alternating bands of calcite and protein. And we take these small cross-sections here. This is about, a centimeter and a half across. And then we can view them under UV light, and we get something that looks like this. And so again, these are alternating bands of calcite, the light bands, and protein in dark, and they're annually produced. So basically, this outer band is the year of collection, and then each subsequent band beneath is around 1 year prior to that. And so because these dark bands are made of protein, we can again do our compound-specific isotope analysis on these bands. So that would look something like this, right? Where you have the outermost ring would be the year of collection, and then the innermost ring would be the oldest date that we have from this coral. So we want older colonies so that we can get longer records of isotopic variability. And these corals can live to be hundreds of years old in some cases. So that's why we're growing or targeting older colonies. Here's just to show you that these are some different isotopic profiles from, in this case we have tissue material, so I showed this earlier from Makkovik Hanging Gardens, and then here we have isotopic ratios from growth bands. So you can see that what is preserved in the tissue material gets passed on to the skeleton. So these are not even actually the same site, but they share very similar trophic amino acid patterns. And basically it means that we can look at the changes to their isotopic profiles through time. Another assumption that we make is that we assume that the corals are eating the best available food to them at that time. And so this is important because, in effect, what we're doing is we're measuring the best available food in the deep ocean with each subsequent year. So if this changes, it's not as if the corals have decided they want to be carnivores or they decided they want to be vegetarians for a while. They seem to exhibit a high degree of selectivity, and they will eat the best food that comes to them. one example of how this is relevant, this is a figure I took from Tessa and her work. She's looking at this question more directly. But we have these different kinds of food webs. So one example could be a more classic food web. You have relatively larger phytoplankton getting passed on to zooplankton, relatively few trophic jumps before we get these particles that are sinking down to the deep ocean. We also have microbial loops where there are more microbes, more small organisms, more trophic transfers, and so we might expect to find difference in the trophic food chain length, which would be reflected in our trophic position calculations that makes it down to our corals. So again, this is not them selecting to different fad diets at the time, this is basically them taking the best quality food that comes to them. So you can imagine that in a microbial loop, this will probably be a more degraded, relatively less fresh food source. So these are some colonies that I worked on that were previously collected. So again, you'll notice, well, I'll go through the different colonies. So we have the years, the time series here. We have bulk delta 50 nitrogen on the y-axis. And in red here, we have an old colony that was taken from a site called Mercer's Monster here in the Hatton Basin. This orange line here is an average of 2 colonies that were collected from Saguelac Bank up here. And then we have one relatively young colony that was collected in 2021 from the Makkovik Hanging Gardens. So again, I hope this highlights that we really don't have a long time series from the Makkovik Hanging Gardens, which is why we really wanted to get there and find these older colonies. And so the point here is that we see this large variability through time in our nitrogen isotopes, and we want to then try and attribute this to other climate records that we can see in the area and other factors going on in the water column that may be explaining this. So this is one example where, say that we compare this to the North Atlantic Oscillation, for example. We can see during positive times we have stronger currents in the ocean. During weaker periods, during negative periods, we have weaker circulation within the ocean. So that the weaker periods would be negative turns in the North Atlantic Oscillation. It's a little bit confusing because they've actually flipped the axes here. But you can see that this negative swing in the NAO kind of looks like it mirrors ours. Positive swing in our nitrogen isotopes. And I'm not trying to say that there is a mechanistic relationship here, but when we see trends like this, we can then probe in deeper using compound-specific isotope analysis. So we could say, maybe this is a change in circulation patterns, and this would be reflected in the source amino acids because you might change the water mass. Maybe there's a change in the phytoplankton community, so we would look at the trophic food length to see if we actually see changes in basically the efficiency with which this food is making it to the deep ocean. And this is an example. We just had some data that we generated basically on Monday. So a lot of this data is just coming in right now, where we can probe in deeper and calculate the trophic position and see if it correlates again with the trends that we see in the bulk isotopic analysis. And so collectively, this kind of highlights 2 of the main pillars that I've talked about so far. So we're very interested in understanding the past, understanding these decadal scale time series in the corals. We're also very interested in understanding the present, and we want to look at the current state of the food web. And so for the past, we were building these time series. Again, we don't have a lot of our samples yet. We've been kind of using coral colonies that were collected from the area and trying to start to understand what's going on, but it's really not until we've got our corals next week that we can start building these time series from the Makkovik Hanging Gardens. We're looking at trying to relate these things to various different climate records. That was just one example, but there's lots of things we want to compare it to. And then we're probing deeper into where there's possibly mechanistic relationships using compound-specific isotopes. And then as for the present, we see these early results hinting at these different divergent food chains at the Makkovik Hanging Gardens, and possibly in situ evidence of the sponge loop. And just a funny anecdote here, this is actually kind of the second time that without trying, my research has kind of been pulled towards sponges. So when I was doing my PhD, I actually gave a seminar here in 2022, and I was talking about some of the interesting geobiological findings that we had from the time. And right around then, Liz Turner had recently found sponges in these Precambrian reefs. And so suddenly, because recently, last summer, I was working on some of these reefs up in the Yukon, and now we're looking for sponges everywhere. So it doesn't seem to matter if I'm working in 800-million-year-old rocks or present Scotian shelf and Labrador shelf. It seems like sponges are always an interesting piece of the puzzle here. And so the last part I just want to touch on is future monitoring. So we're also, in the process of this project, we're building a sclerochronology lab at the d Research Center. The idea here is that after the lifespan of our project, we would love for the community to be able to continue generating samples that they could then send to various labs to continue doing work there. And so we, single-mindedly, I thought everybody would love a coral sclerochronology lab. Very quickly, speaking with folks there and seeing the community, you can see that tuning a lab like this to actually incorporate the fish orbital list. Arctic char there is a really important food staple, so having a lab that allows them to generate samples that are useful to their interests is another goal of this project. And so with that, I'd like to thank, there's a lot of collaborators for this, so I'd like to thank them, and then thank you all for listening.
All right, thanks, Waller. That was terrific. So following departmental tradition, we'll open up the floor to questions. First from students. Give you students an opportunity to ask all the best questions, and then we'll leave it for everyone else after that.
Thank you so much, Waller. That was a great talk. Lots of very interesting science. So I'm trying to calibrate my thinking here because I'm super unfamiliar with deep water organisms. I only really know about terrestrial organisms. So you presented during this talk that the food sources for the different organisms on the seafloor are complementary, and perhaps there's not a whole lot of competition between organisms for certain types of food. And you also said that there might even be entirely separate food webs where some things are eating the traditional fall through the water column versus things that eat this microbial, or cycled food that comes from pyra. So I'm wondering, how much choice do these organisms actually have in what they eat?
Yeah.
And how do we know that they're actually selecting certain foods versus this is just what's available?
Yeah, that's a really good question. It's something that we kind of got a glimpse into in that case study I was talking about. So for a long time, there was this debate whether or not corals in particular are selective or opportunistic. And so we made a strong argument, I think, that the corals themselves are quite selective. And it's really interesting, if you look at the outcrop, they actually are positioned on the outcrop with places that seem to benefit their morphology. So the large fan-shaped paragorgia corals sit basically right at the top of the outcrop and position themselves facing into these really strong currents. So their diet seems to be really linked to their morphology. In contrast to the things sitting in the higher currents, we also see keratosis, these are kind of spindlier corals that sit lower on the outcrop, and basically they seem to be eating like perfectly a one trophic position above. So they might even be eating like vertically migrating zooplankton, very fresh food sources at this site. So in that paper we argued that the corals themselves were very selective. There were some more opportunistic niches, so anemones and a soft coral that we looked at, they were much more opportunistic, more mixed diets. And then the sponges themselves just seem to be filtering whatever they can get. So I would argue that there's quite a lot of selectivity in some of the organisms.
Okay, and then I wanted to tie that into, I guess, what I'll broadly call just ecosystem stress. So you're also studying here what's happening with trawl lines and long line fishing. These are like the anthropogenic ecosystem stresses, but you also commented on, perhaps there's an ecosystem change according to current changes or just sources of food that are coming into this site.
Yeah.
So I'm wondering, what would you expect to happen in terms of selectivity and also in terms of the isotopes that you actually measure under conditions of greater stress? So if you have a lot of trawl lines coming through that blast off a whole bunch of these corals, how would that be reflected in the food?
So the corals themselves provide really critical structure. I would sort of have begun thinking about them again, kind of like trees, where it's not clear that things are directly eating them. Speaking with folks on the Amazon this summer, they sort of say like they're still testing whether or not things are directly eating the corals or just using them basically like structure. So if you think about forests, things aren't necessarily eating the trees, but they like living around the trees. It allows them to get to different food sources. So I think thinking of them kind of like ecosystem engineers is really important. If you raise a forest, what's it going to do to the food web? Well, that habitat's gone.
Right.
And so I think if you do that with corals, the habitat's kind of gone. So that's how I think about it for physical stresses like fishing. When you think about it for climate stresses, this is actually something that Tessa's looking at in her project, but we can potentially see changes in the food web based on the trophic jump. So there's examples where certain phytoplankton communities are moving away from these larger phytoplankton to smaller protists, and we have different metrics for looking at the influence of protists and how that's impacting the food that is making it down. So again, kind of more trophic transfer. So we would consider that to be, in general, kind of a slightly more degraded, less labor food source.
Thank you.
I'm curious, because you said that these corals can grow up to 100, for like up to 100 years or something like that. Is it possible to go back further with these methods?
Yeah. So this is something that I already spent a lot of time doing. Basically, yeah, so if you collect a live coral, you get a year of collection back throughout the entire lifespan. If you collect a dead coral, it may have lived from 1850 to 1950. And so you have to use geochronology to actually find anchor points. So what's nice about the live corals is you know when it died, and it's much easier to calibrate. If you don't know anything about the age, then yeah, it's just this kind of floating time series. One of the ways that we can do that is by looking at the delta-14 carbon. So we can take advantage of that long radio spike in the oceans, where basically we know in the late 50s there's this huge spike in radiocarbon. And so if you can basically find a few different delta-14 carbon points that reflect some part of that curve, it allows you to kind of calibrate where a coral, the lifespan of the coral, where it lived in time. So yeah, that's a really good way to extend the time series back.
Just looking at your cross-section of the coral is really impressive. It's cool to look at. The layers look like they're microns, maybe?
Yeah, they're really small.
Really, really small. So I wonder if you could touch on how do you actually tease apart those protein layers to individually analyze each one with that time series?
So it's really nice that they're alternately banding with calcite. So basically, you take something like this, you plunk it in a little bath of HCl, and so the HCl dissolves away. And so under ideal situations, basically what you pull apart is one discrete year. So the calcite was also part of that year, but we don't really look at the calcite. So we're not sure exactly precisely what part of that year is, but we think we have kind of a time average band. So it's very hard, but that's the general principle. But it's like surgery. You're doing it under a microscope with tweezers.
You're almost peeling like an onion.
Yeah, like a tiny, tiny onion. Yeah.
Very sharp tweezers. Any other questions? Milan, go ahead.
Sure, I'm curious. I think you already implied to Michael a little bit about it, but I'm just wondering at what level or to which degree it makes sense to protect the corals and the sponges and so on. And I can tell you why I'm asking later, but I'm
Sure,
just
yeah.
wondering what do you think?
Yeah, I think the biggest thing is trawl fishing. It's insane how much a misplaced or properly placed trawl, but a trawl can just destroy an entire habitat. And so I think finding thriving habitats, and I think when we find them, we want to look and see that it's cool. There's a lot of negotiations that happen with DFO so that there's kind of some collaboration between fishermen and biodiversity, protecting biodiversity. But if we find high biodiversity hotspots, that's a strong candidate for protecting that site. Oftentimes, there's a lot of variability on the shelf and out to this slope, but it's really the shelf break where these are most commonly found, and these higher substrates where you see the hard substrate that the corals seem to like to. And if you look at our marine protected areas, a lot of them do focus on that site. So the Gully Marine Protected Area, Eastern Canyon Protection Area, they're focused on these sites where there's this high degree of biodiversity. And then sometimes the fishing lines kind of push back on that a bit, but it's a mix between those things.
Yeah, and the reason I'm asking is because we just recovered the instruments in Baffin Bay and like the ocean bottom seismometers, and just deployed them offshore Iqaluit there, and that's at the Hutton Basin Marine Refuge or conservation area was a part of it. So of 25 instruments, we had 6 at the very edge of the Hutton Basin Marine Refuge. And we were given super difficult time about deploying 6 instruments by DFO.
Yeah.
And one of the reasons why we also went to the work in Dark is because there's lots of earthquakes, tsunami potential and so on. We know what happened in Greenland in 2017, people died and so on. So, you know, and we've done nothing, right? So in terms of protecting people. But one thing another reason was that we thought there is no trolling going up, nor because of the maps that we looked, it just didn't seem that there is trolling in Arctic. However, because we use a trawler to deploy the instruments, which is for us rented by DRDC, they basically told us and gave us maps of their, you know, what is being done, and there is a tremendous amount of trolling there.
Yeah.
And what I found really a bit strange is that, because really what, as you said, destroys everything, hundreds of thousands of square kilometers of underwater forest is trolling. And we came with instruments that have less than a square meter footage, and there is 5 or 6 of them, and they're saying, no, they're going in corals there, you can. And we said, okay, well, give us a map, and we'll move them up to 5 kilometers, just we can move them, that's okay, there's no problem. Well, they don't have a map, they just don't want us to. So I was just surprised because what we have, what usually happens in societies, it's black and white. Either if you have a refuge area, you can't do anything. And if you are out of it, you can do anything you want. But there's no common sense. So I had to write that we are actually covering 1/1000th of the 40,000 square kilometers and stuff like that. And then they had to let go because, you know, we basically said, okay, if you look at Iqaluit, it's like a funnel. I've never seen anything like that. If a tsunami happens and there's a large seismicity area, it's going to just run over Iqaluit completely. So I just said, okay, well, it's on you if, you know, we can't learn anything. And then they immediately said, oh, so nobody wants to take responsibility, actually, also there. It's just also black and white. So anyway, so I'm just wondering, do you give input for what is reasonable when you try, like in terms of protection?
I would,
to DFO, like.
If anybody would listen, I would give input. I don't know. I kind of working with DFO, it sounds very much like there's the science side and there's the policy side, and so I think like our role is to produce reports where we identify these hotspots, and hopefully that's taken into consideration. I will say that there's a lot of different types of protected areas. So currently, like the marine refuge, it's not as same as a full-fledged marine protected area. It's less strict, but I think it would be particularly like you can't do bottom contact fishing would be an example. There's this Torngat area of interest with the New Nancywood government has been working to designate as indigenous protected area for many years. They are not necessarily coming up with strict marine protected areas. The New Nancywood plan, they're trying to balance things like safe shipping routes. They want to do fishing in the area, so it's going to be a balance of finding those areas. And an indigenous-led protected area would have different rules than a strict marine protected area. So the hope is that it would, you'd hope that it would have common sense with how people want to live in this area.
Yeah, just because otherwise it's just politics, because we designate something, we say we're great, we're saving, but then we allow totally crazy stuff to happen outside. So it's, it would be much more reasonable to look at what is, how much destructive, and then, but I guess with fishing it's really hard because people just want to eat fish, so. So then, Thanks.
Wilder, we're monitoring offshore Nova Scotia much better than ever, because that's the way science works. And we're learning a lot about the positioning of the Gulf Stream and the Labrador Stream offshore. Do we know anything about the Labrador current and also the what's coming off of southern Greenland in this area on the order of more than a couple of years? Do we know?
Yeah, I'm going to see if I have a better map than that one there. Yeah, this is So the Makkovik Hanging Gardens are actually interesting. So a lot of the water that comes down here is the Labrador shelf water. And so it's a mixture of what's coming out of the Hudson Strait and up from Baffin Bay. And so that's what's kind of flushing over a lot of the shelf here. But the Makkovik Hanging Gardens are this weird, so if you look at, it's this weird sort of basin that looks like this, that's deep for the shelf. And what actually seems to be happening is this Erminger current coming along the slope here, I think is coming up into the basin here. So again, it's really interesting if we're seeing differences in our isotopes. Maybe it's because there's this increased influence of the Erminger. So that's a lot of work that I went did a few years ago, but I'm sort of relying on those results. And we'll see if we can calibrate our results to those and see like if you do see differences in those currents and how it affects food maintenance or not.
I also have a question. I'm wondering why the corals in this area start growing below hundreds of meters. Is it because of the currents and the temperature?
Yeah, so they really they need a hard substrate to basically have it grow fast enough. So they can't grow in sediments, which is why oftentimes you find them on steeper slopes, which is why they're normally at the shelf break. So substrate is huge. Another thing, they do seem to like currents, high currents. They want, like again, they're sessile organisms, they need a lot of food to be delivered to them. So I think those two probably explain why you see them on the areas that we're seeing them in. But yeah, there's different factors that affect where they want to go and their ability to actually recruit to different areas and spawn larvae and something along this kind of a whole other side of it that I'm much less experienced in, that also impacts where they can actually create like habitat.
But is it known what are the maximum depth that they can?
Yeah, so we looked at that a little bit at the end of our last paper, and so very commonly the Paragorgia and the Pronoa, the corals, the big ones I was talking about, they're kind of like 500-ish, 400 meters below sea level. That seems to be their most common distribution. Keratosis gray, the spindly or white ones, they often get quite a bit deeper. And so there's an interesting question as to why that's happening, but they can go to, I think, below like 1,000 meters. I'm not looking at any that go that deep. And then there are other species that I don't know much about, but that can go like way down to the abyssal plain, basically, on things like the around like mid-Atlantic ridge and stuff. There's these kind of different chemoautotropic food sources, so they can be super deep. Yeah.
One quick last one.
Okay.
Do we have bleaching at that depth?
Not that I'm aware of. I think, yeah, I mean, I think that's more of a surface process. So I'm not aware of it happening. The only kind of thing that we sometimes see is kind of necrosis from parasitic organisms kind of tearing away some of the tissue samples, but totally different process.
Okay, as it's now 12:30, I think we'll wrap up there. So please join me in thanking a gift mug for you. So, thanks. Well done. Okay, thanks everyone.